Greetings and welcome to my presentation on biological dispersal and animal home range. We're going to start off by focusing on biological dispersal. So biological dispersal is really referring to the movement of an organism, whether that be a plant, an animal, fungus, away from the original population that it found itself in at birth to a place where they will establish their adult life. One form of biological dispersal is called natal dispersal, which is the first movement away from a birth site to a location, to the location of first reproduction. And then beyond that, there is adult dispersal, where there's going to be a changing of locations to new habitats after this period of reproductive maturity, which might be in between different reproductive periods and also may be representative of movement between breeding sites. Finally, we have gamete dispersal, which is the term used to describe dispersal for non-motile species such as plants. So there are both individual benefits as well as population effects that occur when this process of dispersal happens. On an individual level, dispersal allows an individual to discover preferred habitat, to determine its preferred mating and breeding site, as well as increase its individual fitness. So by finding a region that is preferable as well as beneficial to an individual, that individual is increasing its odds of survival and also of breeding success. On a population level, dispersal has links to population effects through immigration and emigration of a group, while dispersal really describes movement of an individual. The movement of an individual is oftentimes influenced by a group. Colonization is also influenced by individual dispersal, as well as gene flow between populations. There is both passive and active mechanisms of dispersal. On the passive front, which is via mechanisms such as wind and water, this method of dispersal can apply to organisms that are not able to move by their own means. So coral, for instance, is in its adult form is unable to move, but it is able to move in its larval form. So oftentimes passive dispersal will apply to larval organisms because of the fact that they are, while they can somewhat move around, they're largely going to be distributed based on currents. But passive dispersal is often referred to when discussing plant movement of plants. Plants have to distribute something called the disseminule, which is going to be the seed or the fruit, and that's going to be typically dispersed via a natural or an animal vector. So for instance, in this figure to the right, there are several different mechanisms highlighted. In the upper left-hand corner is expulsion, where there's simply just a bursting of the fruit or the seed, the fruit container or seed container, such that the seeds are dispersed. Animals can also eat fruit. That's the purpose or origin behind fruit, and the nuts and seeds and things, nuts, is to attract animals so that they will eat the fruit or the nut, and then they will, thus carry the seed elsewhere. And then wind is another common mechanism that plants use for dispersal, either by having a modified structure that the seed is attached to so that it will catch air, or by having some type of poppy or other thing that will catch the wind. And finally, there's actually a mechanism of water dispersing seeds. The coconut's a good example of that. It allows for dispersal in the ocean from one region to the next. The distance traveled is the result of the velocity and the direction of the dispersal agent. So it is 100% due to whatever the contributing factor is to the dispersal of that seed or that larval form. Active dispersal is going to refer to the movement the active movement by an individual. It can involve flight, swimming, walking, lateral undulation, and typically we refer to that as something that occurs in all stages of the life cycle. So if we're talking about coral, where in the larval form sort of have some form of mobility, we still refer to that as passive dispersal. The individual variation in dispersal, there's going to be individual variation in dispersal difference. So maybe one individual is able to disperse further than another based on its energetic capabilities as well as its musculature. And then size, gender, and other elements can affect dispersal distances. And in this figure to the right, we see kind of an average look at the passive as well as active dispersal distances based on mass, located in the top there, as well as maximum dispersal for those two methods in the bottom panel. The landscape composition and connectivity can actually have an impact on the dispersal as well for organisms. The composition or the land cover type can impact the distance an organism can travel, because if you have a denser forest as opposed to a sparse forest, that is going to limit, or the denser forest is going to limit the blockage or the structures that an organism has to travel beyond. But connectivity, so the distance between different patches of preferred habitat, can also have a big impact on the distance of an organism. If you have a homogeneous region, such as in the upper left-hand set of panels for this particular mallard duck, then there's going to be a lower distance that the organism needs to travel because of the fact that there's such close proximity in those preferred habitats. However, if the connectivity is more fragmented and the patches are more widely distributed, then that organism is going to have to travel or disperse into further distances in order to get to a region of that it can settle in. So there's going to be decreases when there's large distances between those patches, and when the degree of contrast between adjacent types of cover type is going to be large. There's also going to be a decrease in the dispersal when there's a barrier to that movement. So if there's some form of structural barrier or other type of clearing, then that's going to limit the dispersal of the organism. However, increases in that dispersal largely will be possible when there's continuous habitat, when corridors are present, or when there's flows or currents available to assist with that movement. Within marine habitats, we find some other characteristics of dispersal that are slightly different. Firstly, we're very limited in the understanding of dispersal distances because of the fact that there are so many elements within the water, currents, and other things that are responsible for and influence that dispersal. Most invertebrates have broadcast spawning, which is where they'll just release their egg and sperm into the water, and that fertilization will happen outside of the organism. So that initial broadcast spawning can actually have a big influence on the dispersal of the egg and the sperm prior to the larval kind of dispersal that occurs. And the currents are going to largely influence the movement of individuals. So sources of spatial and temporal variation in recruitment are going to be large, in that you'll have this larval production that occurs. The larvae are going to be traveling or dispersing via some unknown distance through some that's really dependent on the bottom topography, the currents in a region, the proximity to any type of underwater communities, and eventually the juveniles will form from fish in particular, and those juveniles will then have their own form of dispersal. So it's very, very challenging, as you can imagine, to really capture all of those different elements of dispersal in a marine environment for a lot of these organisms. For something like a barnacle, however, which is sessile in its adult form, so it attaches to a structure, There's a little bit more limited dispersal that can occur because the larva will have a slight means of passive dispersal via currents. They may be able to have a shorter distance that they travel, so it's a little bit more predictable in terms of the overall dispersal for those organisms. However, again, currents as well as other oceanographic processes may largely influence this the dispersal distances and make it challenging to document the possible range that these organisms can travel. As you can see in this figure, we have the estimated average dispersal distance of the young in miles along the bottom axis. And on the y-axis, we have the number of species and divided into 3 different panels with seaweeds at the top, invertebrates in the middle, and fishes on the bottom. So we can see that seaweeds are going to have a much smaller dispersal because of the fact that they're a bit smaller in size. Their larval forms are going to have no means of mobility, whereas our invertebrates are going to have are documented in this particular 2003 study by Kinlan and Gaines, which was basically like an assessment of all known dispersals of marine organisms. The invertebrates are going to have a much wider range of dispersal, and as you can imagine, that is largely in part due to the fact that there is such a wide range of size and mobility in those invertebrates. And then finally with fish, we find them having more consistently in a larger dispersal within a marine habitat. This is really important to understand because of the fact that some organisms are going to need larger areas in order to ensure their survival. So when you're talking about developing these conservation regions, understanding the range or the dispersal distances of these individual organisms and species is very important for being able to appropriately protect the habitat of these different species. In this figure, we have smaller fish as well as clams and other invertebrates that are located on the left-hand side, which might travel maybe a maximum distance of 500 meters to a little under half a mile. And from there you have larger and larger species of fish, as well as cartilaginous fish, that are going to have a much wider dispersal. So being able to maintain healthy populations of these protected species or target species for fishing grounds really does require being able to parse out an area that's at least twice the size of the minimum area they need. And in order to do that, we need to have some information on the dispersal of these individual species. So how do we track animals? Well, it's important to understand the animal movements, and the movement of an organism from one place to another contributes to things such as habitat use, genetic flow, the home range size, so area requirements for both conservation as well as management purposes, migration routes and stopovers, so understanding the routes that some animals will take in order to move from an area of seasonal regions of resources is important, as well as understanding the occupancy patterns of different animals. So there's a couple different methods by which animals are tracked. One method is using a passive integrated transponder tag. These are small, usually subcutaneous tags that are surgically inserted into an organism. They are going to be useful, but only when they pass a certain transmitter or a radio that is going to pick up a signal from this particular tag. So they really require the animal to be detected within range of the transmitter that's going to receive that information on the location and be able to track it over time. So it's the passive element. There's also a mechanism of tracking animals called marker capture, which is where either a natural characteristic of an animal or a tag that's attached to the animal is used to initially mark it, and then that tag can later be used if the animal is resighted in order to document perhaps the distance that they traveled or the condition of the animal. Same kind of thing on the left-hand side with the dorsal fins of these dolphins. There's specific patterns, and humpback whales are a good example as well. Both dorsal fins as well as the underside of and ridges of whale tails are useful for being able to document the movements of those animals from one part of the ocean to the next. So if there's some notable pattern that can be recognized, then it makes it very easy to have a non-invasive means of tracking and performing mark recapture for these organisms. You won't recapture all of them, you won't recite all of them or see all of the tagged animals, but at least you can get an idea of the population if some of them are recited or some of the tagged animals recited. Radiotelemetry is another means by which we can track animals. This is where there's going to be a base station that's going to receive a radio transmission from a tagged animal. That radio tag will be just communicating as well as sometimes transmitting wireless data to the receiver. And also we have satellite telemetry, which is a means of being able to track these animals via satellite. And with the radio tag as well as satellite telemetry, it's great because of the fact that there are so many various sensors that can be added to these various tags, such as an accelerometer to determine the speed of the animal. There's also a pitch, roll, and other elements that can show the orientation of that animal in space. Additional data from the environment can be collected, such as temperature, depth, things along those lines. And this allows us to put together kind of an overall picture of the movement of the animal and also the kind of conditions within which that animal is moving. Tracking of animals is often scale dependent, and essentially the grain is going to refer to the smallest object recognized by a species. So smaller organisms respond to fine-grained resources or small changes in the composition of resources, whereas larger organisms are going to ignore those fine-grained resources and focus on the more coarser resources. So in a fine scale scenario, which is in this particular example, is dominated by an animal searching around a tree, you'll find a different movement pattern from an intermediate scale, where the dominant movement is going to be among forest patches. So as we are zoomed in on the left-hand side, we can evaluate the movement of that animal in a different light than at this intermediate scale. And finally, on a coarse scale, we see that the movement of animals between different forest hills or different forests is going to occur at a much larger scale and thus give us some information regarding the viability of these different forest regions or patches. And within this figure, we have a couple different panels. In the upper left-hand side, we have and overall, we have a spatial autocorrelation in the changes of resource availability. So in the upper left-hand panel, we can see that there's a link to frequent and shorter movements of animals when there are fast fine-scale changes in the resource availability. In the upper right-hand corner, we see that there are frequent longer movements when there is fast large-scale changes in the resource availability. And along the bottom, we see there's somewhat the opposite, infrequent movements of the organism when there's slow fine-scale changes, and infrequent movements of organisms and longer movements when there's large-scale changes. So the changes in resource availability is going to have a large impact on the scale of movement as well. A good example is a study regarding the wandering albatross. This study was conducted by Fritz et al. In 2003 and incorporated or looked at breeding at the Crozet Islands, which is the only period of time where these birds actually contact land. So if they're not breeding, which happens at reproductive age of 5 years or older, and then thereafter every other year. So if they're not breeding, then they're actually just going to be foraging over a very large area of the ocean, approximately 2,500 nautical miles. Oftentimes, this ocean region is going to appear homogeneous and lack topography as well as structures. So what would the movement paths look like for this particular organism? And this study took it upon themselves to describe that and look at some of these movement patterns. So there is quite a bit of heterogeneity in the ocean. Upwelling along the coast is going to result in productive waters and more food availability for these organisms, and that's closely linked to the bottom topography as well as the coastal conditions. The wind is going to be an important element for helping to drive the movement of these organisms. They need to have favorable winds in order to conserve energy while flying. And then swells are going to be responsible for creating updrafts that give them lift. So if we take a look at something called wind shear, we can see that in this figure, the wind speed is going to increase with increasing altitude. And these albatross can leverage that. As you can see in the portion of this figure to the right, the strongest winds are going to be higher up in latitude. So at 39 meters or so is where you're going to find the fastest winds. So the birds are able to utilize that layered kind of wind speed to their advantage when moving from one part to the other of an ocean environment. So in the study approach, they tagged 11 different albatross and collected information on the location, the GPS location of these animals at 1 second intervals. They only evaluated a minimum of 5-hour tracks per bird. And out of that, they used a mechanism called fractal analysis. This is where a fractal value of D is going to be used in order as a mechanism for measuring the tortuosity or the twisted or turning nature of a path. The fractal D value is going to be derived from the relationship between the line length and the set of dividers, or the distances that are used to calculate it. So the divider size is going to be really important, and you're going to evaluate the path at a certain distance and look at how many turns or how many twists and turns occur along that path of a certain distance. A value of 1 is going to be representative of a straight line. So in the bottom right-hand figure, we have divider size along the x-axis, and we have the fractal D measurement along the y-axis. A value of 1, as you can see along the bottom of this scale, will be a straight line, whereas a value of 2 is going to be so tortuous that or so twisted that the path actually folds back onto itself. So in the figure to the left-hand side, you can see a complete path of a bird, the tracks of a bird over the period of time from this particular animal's journey. And we can kind of get an idea of where those periods of higher tortuosity are going to occur just by looking at the overall path. In this figure, we see the path of all the 11 birds that were tagged. We can also see that all the birds exhibited at least one major change in the fractal dimension, or the D value, at a divider side of about 100 meters. So right about here, there's 100 meter. There was at the 100-meter mark, there was a large kind of shift or increase in the fractal dimension value, which suggests that there was quite a bit of twisting that occurred within the path of the animal. There was also evidence of a secondary kind of transition at a divider size of about 10 kilometers. So if we look at the divider sizes in terms of the distances where there are shifts, we've got one kind of section that occurs between 0 and 100 meters, another division that occurs between 100 meters and 10 kilometers, and then final bit of variability in the path there. So in terms of the results, the tracks varied from 149 to over 1,000 kilometers in length. There was that transition at 100 meters and 10 meters, which created several domains that were evaluated in this particular study. Within those domains, the first domain had a overall value of 1.01, which indicated it was very straight and indicated an adjustment to wind during the foraging stage. In the second domain, there was a less straight path that each of the birds took, which indicated or suggested intensive use of the foraging patches. And then the third domain made use of flyways with favorable winds for traveling longer distance between the colony and different foraging patches. So if we take a look at this figure from a single bird, we have that information on the fractal dimension, or the D value, kind of divided into these different domains, with transitions occurring at the 100 meter and the 10 kilometer. And as you can see, there is, as you can see in this figure, there's quite a bit of variability in the movement from a finer scale movements to larger scale movements of the organisms. So the fractal dimension, or the D value, is not consistent, and it is scale dependent in these birds, is what the study found. So home range estimates is an important element of being able to understand the movement of organisms, or something to take away from that movement. A home range is a region that encompasses all the resources that an animal requires to survive and reproduce. It's also going to be an area where the animal is going to spend the most of its time. This may include territories or defended space, but a home range is not going to be all territory. There is going to be some of it that is not a protected or defended portion of the organism's habitat. The species grain size is not really an exact measure, but there tends to be a positive correlation between the extent of the organism, extent of the home range, and the grain size of the species. So in this particular set of figures, on the left-hand side, we have a coarse-grained kind of scenario wherein there's going to be a shark species that uses a large area of the Pacific Ocean. And in the right-hand side, we see a species that is considered fine grain. This is the range of the vaquita porpoise, wherein, if you see in the red box there, is the refuge, which is in a very small portion of the Gulf of California, adjacent to Baja California. And if you take a look comparatively on the shark map, that's going to be in this little protected region, whereas the shark distribution, or the shark home range, is going to be quite significantly larger and more coarse-grained. So in order to estimate the home range, we need to know locations of individuals over time. We need a method to convert that point-based data into an estimate of area, and we need an idea of the areas that are likely to be used. One mechanism of calculating that home range is the minimum convex polygon method, or MCP method. This is an easy to compute mechanism by taking coordinate data and turning that into a kind of general region or space where the animals may exist. There's no intensity of use here, so there's only the location information. There's not any type of probabilistic interpretation here. And a space is either included or excluded. And then finally, a disadvantage of this is that it likely is going to include large areas that are not going to be used by the animals. So in this particular figure off to the right, which is indicating the buffered MCP for herds, forgetting what kind of herds, but the herds are, the points of those herd locations are found within that polygon. However, there's some spaces where we might question if they should be included in the overall kind of home range of this organism, given there's not anything occurring in this big space here. Kernel estimation, or the kernel home range estimation, is another more kind of involved process for estimating the home range. In this particular method, it's based on the probability of distributions. It uses a surface density, which is generated to inform the likely distribution. And this happens by having components or different sightings, again, location information of these animals, and also incorporating things like the number of individuals that were found in a certain region. All those components are then used in order to determine a density value. And that density value over the suite in the x-axis here, you have all of your different quantities or I guess numbers or points of those different sightings, and you have density off on the y-axis here. But they can be converted into this surface density model over on the right-hand side, which looks kind of like a heat map. This can be used to determine core areas. So if there's more sightings within this particular region of the map, then we'll have a brighter kind of scale to that, and thus we can probably conclude that this is going to be a region where there's a higher sighting level or higher use for the organism. And just to give you a comparison of that MCP, which is represented by the lighter kind of polygon shape here, and a 95% kernel density, so where 95% of the population is expected to exist, represented by the shaded area. You can see that polygon, which is a little bit challenging to see given the fact that it's sort of kind of overlaying these other two polygons. But you can see that there's a large space here wherein that MCP polygon is really not representative of the use of these elephants, the habitat use of these elephants. So within that kernel density, we see that there's a much better representation of that space that's used by the elephants. However, in this particular figure, the dark bolded line is the preserve area that was decided upon for this particular organism. So there's a few influences on the home range size for organisms. Animals tend to use an appropriate size area that meets their needs. This is typically determined by their body size, mobility, and social interactions. And then the distribution and intermixing of resources is going to be an important influence on this home range size as well. Animal behavior is going to be large influence on this home range distribution. For instance, the influence of a predator can have a big influence on the prey density within a region. In this particular figure, we have the green line representing the occurrence of that particular deer. And then the predator lines are represented in the red lines. And you can see that in a region where there's an increase in the predator population, there's going to be a decrease in the deer population. You might also see the behavior of herd preference within certain habitats. In this caribou herd region kind of distribution, in the top two panels in the bottom left, we see that there's a difference in the dispersal or the home range of each of these herds to allow them to not have to compete for resources. They've kind of distributed themselves in different parts of this particular portion of Alaska. There can be environmental influences on the distribution of the home range of these organisms. For instance, in this figure, we have the decreased movement located to the left of the panel, increased movement to the right, and we see that there's a difference in the amount of precipitation as well as the temperature in respect to how these various species are going to move within a particular habitat. So when there's there's a decreased movement when there's lower temperatures within the winter as opposed to summer, and there's a few other things we could probably work out from this. Food proximity is another important factor. In this study by Velix, 2012, they looked at the relationship between water hole dispersion, where there is likely to be prey present, and the lion home range size. And we can see on the x-axis that the mean distance between two neighboring water holes, as that increased, it increased the September home range size of the lions. So with larger watering holes, it means there's a greater distribution in the prey, and thus the lions have to follow that prey. Finally, there are paths to different resources that animals can take. We find that there are migratory movements that a lot of species will pursue in order to take advantage of seasonally available resources. In this figure to the left, we have the flight paths of various migratory birds around the world. You can see they're highly mobile and they're going to travel huge distances. And on the right-hand side, we see the migratory paths of blue whales from their feeding grounds in the blue to the breeding grounds in the pink. And there's huge distances that they travel in order to go from one region to the next. However, it's important to note that barriers to these migration paths are detrimental because animals cannot detect those changes. They're not able to determine those large-scale distance or changes in the environment or the corridor that enables them to get to these different breeding grounds or feeding grounds. So that can be have a huge impact on a lot of these highly migratory species. And that's all I have for you. I hope you've enjoyed this presentation.